Welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. So good afternoon everyone and welcome to the July FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. Last month we heard from Dr. Beck Sheen, who's our very own FightMND Director of Cure Research and Programs. Dr. Sheen presented to the research community our incoming FightMND cure and care research strategies, outlining how it will drive our investments over the next 5 years. If you missed out on this talk, it is now available on our website, so you can catch up at a convenient time. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you are using the app-based version of Teams, and unfortunately it does not work in the web-based version. Please do ensure you introduce yourself and where you're from before asking your question. So now to our speakers for today. Associate Professor Kelly Williams is the Head of Genomics and Bioinformatics for the multidisciplinary Macquarie University Centre for Motor Neurone Disease Research in New South Wales, having been recruited as a founding member of the centre. Her research has been supported by MND RA and NHMRC fellowships, and she leads a team that has been instrumental in breakthroughs for MND and frontotemporal dementia gene identification. She's joined today by her postdoctoral fellow, Dr. Lyndal Henden. Dr. Henden completed her PhD at WeHi in 2017 and joined Associate Professor Williams' lab shortly after. She's a specialist in the statistical analysis of genomic data, and the methods she has developed have been successfully used to identify loci containing disease susceptibility genes. Associate Professor Williams and Dr. Henden join us today to discuss their latest research into the genetic causes of MND. So over to you guys. Thank you.
Thank you, Sarah, and thank you so much for the invitation today for Lyndal and I to be able to talk about the importance of genetic research in motor neuron disease. So I know we just had a very lovely introduction from Sarah, but I thought I'd just give you a super quick brief one about us and about where we're from. So I'm Associate Professor Kelly Williams and I'm a molecular geneticist, and I've been working on motor neuron disease genetics for the past 16 years. So initially at Concord Hospital and then moving to Macquarie University in 2013 as one of the founding members of the MND Research Centre. And so my senior postdoctoral fellow, Dr. Lyndal Henden, is a statistical geneticist, and so she has a background in mathematics and statistics. And she has been at Macquarie with our team for the last 5 years after she completed her PhD down at WeHi in Melbourne. And so, together we are leading a genomics and bioinformatics research program within Macquarie Medical School, making some really exciting discoveries. And so we are part of the multidisciplinary Macquarie University MND Research Centre, which is a collection of 8 different research teams made up of a diverse range of expertise from academics, academic researchers, professional staff, alongside the multidisciplinary neurology clinic, which is located on site at Macquarie University. And so we are a very diverse range of expertise that work together to try to answer some really, really important questions in motor neuron disease. And what you can probably see here, so we have our different research teams, we have our clinic, and at the centre, so at the centre of our centre, effectively, or the core of our centre, is our patient biobank. And this is really one of the most, I guess, critical aspects of our entire research centre. So the patients, MND patients, are really at the centre of what we do. So we have the largest biobank of its type in the Southern Hemisphere, and through participation in this biobank, people with lived experiences of MND, so PLEX, so patients, carers, their families, although they make very important contributions to MND research through philanthropy, advice on strategies for research projects, understanding what the most pressing needs are for patients. In practice, their participation in the biobank is really what facilitates our research and is a way for them to make really meaningful contributions to research. And so I established the Macquarie Neurodegenerative Disease Biobank back in 2013 when we relocated to Macquarie University. And the main rationale was that for genetics work in particular, we really need patients. We need patients, we need family trees, we need samples to be able to do incredible genetics work. And so in the last 10 years, this biobank has now grown to have more than 1,000 participants, so MND patients, control participants, with more than 60,000 longitudinal biospecimen collections. So as I mentioned, this is the largest biobank of its kind in the Southern Hemisphere and the largest MND specific biobank collection site in Australia. And this not only facilitates a lot of obviously our genetics research, but many of the other research teams within our research centre and throughout Australia. So one of the most, I guess, unique things about our biobank is the fact that we have longitudinal sample collections. So because our biobank has been around for a decade now, we have lots of participants who have donated samples and who have since become deceased. So we have full sample collections from onset all throughout disease duration until death. So longitudinal clinical, demographic, biospecimen collections, including DNA, RNA, plasma, serum, urine. And then for a subset of patients, we also have post-mortem tissue obtained after death. And so having this really large data-rich cohort has given us further insights into clinical heterogeneity in motor neuron disease. And I will use ALS and MND interchangeably throughout my talk. And so clinical heterogeneity is where we see differences in disease. So MND is complex. We don't always see, there's nothing that we would, I guess, consider a very standard disease. So we can, in our biobank and our sample collections that we hold at Macquarie, we have more males present with motor neuron disease than females. However, if you are a female, you actually have a worse prognosis than males. And we suspect that this is because that there are more females, the proportion-wise, more females have bulbar onset motor neuron disease than males. And we do know that bulbar onset MND also has a worse prognosis. And so we can see in the slides here, this is just, this is a clinical summary of more than 600 MND patients. We have variation in age of onset. So some patients will present at the clinic in their 20s, others in their 90s. We also see a range of disease duration. So we can have patients who live for 2 months, patients that live for 2 years, and patients that live for 2 decades. So this is what we see in Australian MND patients is actually quite comparable to what is seen around the world. In the figure on the right-hand side, we see this is a diagram describing ALS-FRS, which is the ALS Functional Rating Scale, which is it's a measure used by clinicians to be able to demonstrate the progression and deterioration of a patient living with MND. And so even within this, we see heterogeneity. So although the ALS-FRS score trends downwards over time, for some patients we can see a very rapid decline and then it's steady off. For others we see steady and then a severe drop off. For some we see a nice linear decline. For other patients, you actually see an increase before you see a decline. So again, it's just, I guess, highlighting the heterogeneity. we've tried to use trajectory clustering models with our machine learning collaborators and statistical collaborators to try to, see if we can categorize the progression into subsets, and it doesn't seem to exist. So we're seeing all the time such a large heterogeneity within MND, further adding to its complexity that we already know about. So when a patient comes to our clinic, we will define them or categorise them as either familial motor neuron disease if we see a clear family history of disease. So this is where MND is being inherited from generation to generation, or we classify them as sporadic where there's no clear family history of disease. However, this binary classification, it's quite useful for research purposes and also in the clinic, but in reality, it's not actually what we see. So again, was just adding complexity to MND. We see this spectrum of inheritance. So we have low penetrance families. So in these cases, it looks as though MND is actually skipping a generation. Or we have patients that present at the clinic with sporadic MND, so no clear family history, but then we find that they actually have a gene mutation that's known to cause the familial form of disease. And so a lot of my research had originally focused on the familial form of the disease, but over the past 5 years has really spread across this full spectrum of inheritance. So over the last 30 years, since the first gene was discovered to cause MND, so that was the SOD1 gene back in 1993, in the last 30 years, we've identified more than 30 genes as either causing motor neuron disease. So these are ones that are found, inherited in families, or genes that are associated with motor neuron disease. And so I have a few figures to show gene discovery over time, and the reason I have a few is that there's actually no perfect figure. There's no perfect figure, so they all show something a bit different. So this one that I have up now is the most recent version, which describes the genes that have been discovered in MND over time. This is probably my favourite figure. However, it is 7 years out of date. But what it shows here is the increase in genes attributed to the familial form of disease. So starting in 1993 with the discovery of SOD1, and we can see a steady progression and then an uptick of gene discovery, which occurred with the introduction of next generation sequencing technologies, which really accelerated gene discovery in human genetics research. And so even though this is slightly out of date, this figure, we can see by the end of 2016, around 70% of familial ALS cases have a causal gene attributed to their disease. So one of the, I guess, a clear outcome or benefit from finding disease genes linked to ALS is translation to the clinic. And so when we find disease genes or find genes that we know cause motor neuron disease or associated with motor neuron disease, they can be added to diagnostic panels. And so this means when we have patients coming to the clinic that have a suspected family history of disease, we can get a genetic diagnosis in around two-thirds of those patients, which can be really important for future decision making, which could include pre-implantation genetic diagnosis through IVF. So this is where a gene mutation is known in a family and individuals choose to go through IVF to avoid the mutation being inherited by the next generation. Very excitingly is when new genes are discovered, it provides the opportunities to develop gene therapies and therefore allows individuals to be involved in clinical trials for these gene therapies. And probably the most exciting one, one of the most exciting things to come out in the ALS space in a very long time was in April, the FDA approved a gene therapy, tofersen, which is an antisense oligonucleotide gene therapy against SOD1. So this now provides a therapeutic treatment for MND patients that have tested positive for a mutation in SOD1. It also now means that they're for pre-symptomatic carriers. So these are individuals that carry certain SOD1 mutations may be eligible to undergo a clinical trial as a pre-symptomatic patient. So they have not developed motor neuron disease at this current time. And so another major outcome from gene discovery is actually allowing researchers to understand the pathological mechanisms underlying motor neuron disease. And here is another one of these figures. This one, unfortunately, is only from 2015. But what it shows here is when we find gene mutations, we then learn about what the biological function of those normal proteins are. So when a gene encodes a protein, when we have a defective gene, it can lead to a defective protein, and we can then, the functional biologists can understand what the actual mechanism is happening within that defective gene. And so gene mutations over time have allowed us to, I guess, converge on common functional pathways. So a lot of these genes will overlap in biological pathways or functional mechanisms to further understand what's happening in MND. And so I wanted to give an illustration of at our MND Research Centre at Macquarie, how we are, I guess, following this research pipeline from gene discovery towards a therapeutic treatment. And so in this figure here, you can see just generally how all our research teams work together. So our research feeds into other teams of research and backwards, and you can see in the middle, at the core of our centre, which is our biobank, how critical this is participant samples feeding into each of these different research streams within our large research program. So as an example of the research pathway, in 2016, I found causal mutations in an Australian ALS and FTD family in the gene CCNF. And so this then led to prevalence screening, so looking at the prevalence of these mutations in Australians and elsewhere throughout the world, including correlating the genotype with phenotypes. It's led to genetic counselling implications for individuals in the clinic. The gene discovery then goes into our functional biologist team, who they really try to understand biologically what is this defective gene and therefore defective protein doing. And then we can end up into animal models. And so specifically for CCNF, so the protein that it encodes is called cyclin F. The functional biologists at Macquarie, they determined that this mutation leads to a defect in neurons, which leads to an accumulation of a neurotoxic protein. So they then went to develop a gene therapy, a potential gene therapy that actually negates this protein accumulation. And so there's a spin-off company out of Macquarie University called Celosia Therapeutics, whose purpose is to support the potential gene therapies that are developed at our university, to support them through to clinical trials. Specifically, this one of the examples is this cyclin F gene therapy. So I guess I just want to use this to give an illustration of this research pipeline, like why we try to find genes to ultimately end up at gene therapies. And so now I'd actually like to move on to some of the genetics work that we've done. So this pie chart here really summarises 25 years of mutation screening following gene discovery in Australia. So we can see that more than 50% of MND families in Australia have a mutation in either the C9orf72 gene or in SOD1. And in brackets is the number of Australian families with these mutations. And so I'm going to focus on SOD1 just for a moment. So this as I mentioned, this was the first gene that was found in 1993. And since then, we've had 34 families have their disease attributed to a mutation in SOD1. In Australia, the three most common mutations are the I114T. So this is a amino acid at position 114 in the protein and the amino acid that it's mutated to. And so these three mutations make up around, I think 25 families. When Lyndal came and started working for us 5 years ago, one of the first things she did was actually look at genetic ancestry of all of these SOD1 individuals. And what we were able to find was that these, even though we had 34 families here, many of these actually came from a common founder. So a specific example of our I114T families, 20 families we found were actually one big mega family that had originated from a common founder in Scotland. So I'm not going, as much as I would love to talk about the cryptic relatedness work that we're doing, that's it. We will move on to some more genetics. So in our familial MND space, we have lots of patients that have identical gene mutations, so within families or across families, but we still see clinical heterogeneity. So we're still seeing this variation in the presentation of disease, so the site of onset, the age of onset, the disease duration. And so a study that a postdoctoral fellow, Emily McCann, conducted during her PhD. So she and I compiled an extensive clinical information across all of the patients that we genotyped that we knew had mutations known to cause MND. And we saw significant differences within the clinical features between these different mutations. So in the figure on the left-hand side at the bottom is age-dependent penetrance, which I will talk more about in the next slide. But we can see here that these 5 different SOD1 mutations all have different varying degrees of age-dependent penetrance. In the middle graph down the bottom, this shows the survival. So this shows that the length, the time that a patient will live after onset of symptoms with a specific SOD mutation. And we can see, again, there are, across these 5 different mutations, some of the mutations have a worse prognosis than the others. And then in the figure on the right, this again just shows the differences in disease duration of patients with these 5 SOD1 mutations. So red is what we would consider very rapid motor neuron disease, so disease duration less than 2 years. Orange would be considered, I guess, average or standard disease duration, so between 2 and 5 years. And then yellow is long, what we would consider long disease duration in MND, so longer than 5 years. And again, we see variation across mutations, but also within mutations. Now having a look at C9orf72, which is the most common genetic cause of MND in Australia, in both familial and sporadic cases. And age-dependent penetrance, it's always it's something that comes up quite often when we're talking to patients or in genetic counselling information sessions, and it's to do with the probability of what age you would become affected with MND if you carry a mutation. So to describe this figure in more detail, in an example, if we took age of 70, we go up from the age of 70 to where we hit the age-dependent penetrance line across to the cumulative frequency percentage. So in this example here, for females that have a C9orf72, a pathogenic C9orf72 repeat expansion, at the age of 70, we would expect that around 2/3 of mutation carriers would be affected, but that means 1/3 of carriers are not yet affected at the age of 70. For males, males have a higher age-dependent penetrance. So at the same age, at 70, a male who carries a C9orf72 expansion at the age of 70, more than 80% would be affected with MND. We also we perform the same analysis with sporadic cases that have a C9orf72 mutation, and we didn't see any difference between familial or sporadic cases. So they're still showing the same age-dependent penetrance. But again, what this means is that if you carry a mutation that causes disease, this is a statistical calculation of at what age you may become affected. So you may become affected early, but you also may not become affected until very late age, which is probably to do with more genetic factors that we have not found yet. So these are our familial mutations, but what about genetics in sporadic motor neuron disease? So you would remember my figure at the start that in familial MND, or even in reduced penetrance MND, we see family history of disease. MND is occurring in more than one individual within a family, whereas in sporadic, it seems to be an isolated incident. But many studies over the past, probably 15 years have shown that genetics are important in sporadic MND. And one of the statistical methods we use to demonstrate that is called heritability estimates. And they, just as a small example, it can use the concordance between identical twins, the concordance of disease between identical twins and non-identical twins, looking at risk ratios of families. I don't want to get into it too much, but it's basically showing that sporadic MND is not definitively caused by the environment. There is a genetic component in around 50% of sporadic MND. So these genetic component may be risk alleles alongside environmental risk factors, but it's showing that genetics contributions are important in sporadic MND. And also what I showed before is that we can have sporadic patients present at the clinic, but they actually have mutations that are known to cause MND, such as C9orf72. So over the last 30 years, more than 1,000 genetic variants have been reported in ALS-MND in the literature, which is a lot. And so we wanted to say, how many of these genetic variants, of these 1,000, how many of them are actually present in Australian patients with sporadic ALS? Is this a very important aspect of sporadic ALS in Australia? So in this study, it was a very, very comprehensive study led by Dr. Emily McCann and Dr. Lyndal Henden, where they compiled all of the genetic variants across all of the literature. So 546 publications they reviewed to generate, at the time of this publication, a list of 850 genetic variants that have been either causal in ALS, in MND, or associated with disease. We've now updated this list to more than 1,000, but for this publication it was 850. So they effectively generated a catalogue of all of these genetic variants, which didn't exist to this extent. So we were the first ones to do something to generate this catalogue to this extent. And for each of the variants, so for each of these 850, now over 1,000 variants, we generated additional annotations and additional information. So genomic locations, transcripts, including nomenclature corrections where the actual mutation was reported incorrectly in the paper for the position and the position that it was located at. We also provided allele frequencies. So this is the prevalence of this variant, this genetic variant, in a control population, and which is important to understand if it's common in a control population or not. Also the country of origin that it was first reported in, and this can be really important because cross ancestral groups is differing prevalence of causal gene mutations in ALS. So as an example, in MND patients of European ancestry, there, as I mentioned before, there is a high proportion of C9orf72 mutations and a high proportion of SOD1 mutations. In direct comparison to those MND patients of Asian ancestry, there is almost zero C9orf72 repeat expansions, and they have a much higher proportion of SOD1. So this was really important to put together in our catalogue. To give you an idea of, I guess, how many mutations or gene variants can be out there. So here, this is the DNA sequence for SOD1. So it's 460 nucleotides, and which make up 154 amino acids. And in this one gene, there are 197 genetic variants across SOD1. So this figure shows that the rectangle at the bottom is representative of the full gene. And these are all the gene mutations that have been reported in the literature in ALS in SOD1. And so we generated this catalogue, and then we looked across all these 1,000 or 850 and then 1,000 variants in whole genome sequencing data from more than 600 Australian sporadic patients. And so, as I mentioned, we published this a while ago, but what we found was, of these 1,000 variants, there was 43. There was only 43 of these variants found in sporadic cases. Now, remember, a lot of these variants may have been reported in the familial form of the disease. So here is a summary. That's the thing with genetics work. You can so much of your results can just be displayed in a simple table. So here were all the genes and the variants that we found in individuals. And I'm just going to see if I can pull up a pointer. No, I can't. So that's all right. Nope, that's all right. So what we see, there are singletons that have mutations. So these genetic variants occur in one person, or they may occur in a lot of people. So maybe I can get, here we go, laser pointer. So we can see C9orf72, we actually had 41 sporadic patients that had this causal mutation. We also had 3 patients that had a known SOD mutation. And I'll get rid of this. And so what we found was that at least a third of our sporadic patients had at least one reported genetic variant, but 7% had 2 or more. And this is what we would consider either polygenic or oligogenic inheritance. So oligogenic inheritance is where you would have a causal mutation, such as C9orf72 or a SOD1 mutation, and then you have a second or a third genetic variant that is associated with disease. So not necessarily causal, but statistically associated with disease. And we found many patients that had this. We also have polygenic inheritance. So these are when patients may have more than one associated genetic variant. So not necessarily one that we know to cause MND, but associated with MND. And then when we looked at the clinical data of these, so do genotype, phenotype correlations, we found that these patients that had either oligogenic or polygenic inheritance, so more than one genetic variant known to cause or associated with MND, they actually had an earlier age of disease onset. And so, we generated this fantastic catalogue, and so we may as well make it useful for not only the MND research community, but also clinical community. So our data was used for the major update of ALSOD, which is the ALS online database. And this is managed by our colleagues in King's College London. And it's basically a web-based browser, a database that outlines all the genes that have been implicated in ALS in the literature and all of the variants. So it hadn't been updated for quite a long time. We'd put our data set together and it was used for the major update here. So this, as I said, this is used worldwide by researchers and clinicians. So, a way that Australian researchers were contributing to global genetic efforts. But as I mentioned before, more than 1,000 had been implicated. We only found 43. And we found more in our familial form, but not 1,000. So how do we know that these genetic variants are leading to disease? And this is another major large initiative that we are involved in, and it's called ClinGen. And it's a initiative out of the US, funded by NIH, which they look to obtain evidence from the literature, so from medical research literature, to actually understand the clinical relevance of genes. So we're involved in this as curators, so as actually collating the evidence to determine whether these genes are definitively associated with disease, or maybe there's only limited information available. They could have been discovered 20 years ago, before next generation sequencing, when maybe reporting thresholds were not as strict as they are now with our large control databases. And so we've been involved, as I mentioned, we've been involved in this as curators as well as gene experts. And so this is just looking at genes, And what we hope to move into, which I think is planned for the end of this year, is to actually extend this to start looking at variants. So the first gene that we'll be looking into as part of ClinGen will be SOD1. As I mentioned, there were 197 variants that had been reported in the literature in SOD1. So we'll be going through all of these, all 197, to see which of them actually have strong evidence that they are causing motor neuron disease. And so that's it for me. I'm now going to pass over to Dr. Lyndal Henden to talk about one of our other really exciting studies that we've recently published this year. Thank you.
All right. Thanks, Kelly. Let me just share my screen. All right, so I am going to be talking about a complex form of genetic variation and the sort of clinical and genetic heterogeneity that we see with this. And this is all to do with short tandem repeat expansions in sporadic motor neuron disease and frontotemporal dementia. So a short tandem repeat expansion is a repetitive sequence of DNA. So for example, we have this CAG motif, and it can be repeated consecutively multiple times. And these short tandem repeats, or STRs, they have a high mutation rate. And so they can either expand or shorten when they mutate, and this can lead to disease. So for example, in the gene ataxin-1, most healthy individuals will have less than 34 CAG repeats. However, individuals with spinocerebellar ataxia type 1, they will have more than 38 of these CAG repeats. And similarly, in the Huntington's gene, most healthy individuals will have less than 36 CAG repeats, but those with Huntington's disease will have more than 39 of these CAG repeats. So these STR expansions, they cause up to 50 diseases, and this includes over 20 neurodegenerative diseases, including Huntington's, a number of different spinocerebellar ataxias, and this hexanucleotide repeat expansion, this 4 Gs and 2 Cs, in C9orf72, that is the most common cause of both ALS and FTD, or motor neuron disease, and FTD, excuse me. So these expansions, it's really interesting because they can be associated with multiple diseases. So for example, the C9orf72 repeat expansion can cause either motor neuron disease or frontotemporal dementia. So in healthy individuals, they tend to have less than 30 of this repeat motif. However, individuals with these diseases tend to have hundreds to even thousands of these this hexanucleotide repeat expansion. And C9orf72 is not the only gene where the same repeat expansion can cause different diseases. So in this Notch2-NLC gene, a CGG repeat expansion causes this neuronal intranuclear inclusion body disease, but it's also been found in individuals with multiple systems atrophy and OPDM. So the same repeat expansion can kind of cause different diseases here. And it's also really interesting that the number of these repeats can be associated with different diseases. So, for example, individuals with spinocerebellar ataxia type 2, so SCAR2, they have more than 33 CAG repeats in a gene called ataxin-2, and that's causal for their disease. But individuals with sort of 29 to 32 repeats, that repeat length has been associated with motor neuron disease. So it's a risk factor for motor neuron disease. So the same repeat expansion, just different sizes in the same gene, has been associated with different diseases. And the same thing is true for a CGG repeat expansion in the FMR1 gene. So more than 200 of these repeats causes Fragile X syndrome. However, a shorter number of these repeats is associated with Fragile X tremor/ataxia syndrome. So it's slightly different. And when we see sort of these slightly shorter repeat expansions, it's sort of this grey area, and we call this intermediate expansion. So there's a number that you would typically see in healthy individuals, and then there's a larger number that you typically see in individuals with a disease, and then you kind of get this grey area where there's a kind of, it's bigger than what you see in normal individuals, but shorter than what you see in individuals with a particular disease, and that intermediate zone can be associated with different diseases, different clinical symptoms, even sort of reduced penetrance in families as well. So it's quite complicated, these short tandem repeat expansions. And they're typically identified kind of using wet lab techniques, so PCR or southern blotting, and this can be quite time consuming and expensive. If you have really large cohorts and you're wanting to screen a number of short tandem repeats in that cohort, it's not really scalable when you're using these wet lab techniques to be able to do this. And so there hasn't really been many comprehensive studies of many short tandem repeat expansions in large disease cohorts. However, with the emergence of whole genome sequencing data, it's really sort of changed the way that we're able to look at these STR expansions, and we're able to do this now with really nice and fancy bioinformatics techniques. Whole genome sequencing data and some clever statistics allows us to now analyse the really large disease cohorts for a number of short tandem repeat expansions, just to see what's out there and if there might be things associated with disease that we wouldn't typically screen a disease cohort for. So our aim was to screen sporadic Australian MND and FTD patients using bioinformatic methods for quite a number of STR expansions, just to see whether or not they were overrepresented in our cases versus a control cohort. So we have recently published this work. So if anyone wants to read it in more detail, it's come out in Science Advances earlier this year. So I'm going to show you some of the results from this study. So we had whole genome sequencing data for 608 sporadic motor neuron disease patients, 68 sporadic FTD, and just under 5,000 control individuals. And we used a bioinformatics tool to screen for 21 neurodegenerative disease-associated STRs. So this includes we screening our MND and FTD patients for the Huntington's expansion, for expansions that cause myotonic dystrophy, and a number of spinocerebellar ataxias. And anything that we found that was above the normal range, we went away and we PCR validated to understand whether or not it was a truly expanded repeat, and if it was beyond what you would see in a normal individual, if the length of it was either this intermediate or pathogenic size. So we found that 22% of patients have a repeat expansion beyond the normal range. We found it in 10 genes. So I just want to explain this picture a little bit. So these are the 10 genes where we found an expansion beyond the normal range using our bioinformatics techniques. So I'm just going to, maybe if you just look at spinocerebellar ataxia 8 here, for example, we've got the blue block, the yellow block, and then the red block. So those are literature-defined sort of boundaries of what's considered a normal length versus the intermediate grey zone versus what's usually pathogenic for SCAR8. And we ran everyone through our bioinformatics tool, and we got the number of repeats for that particular disease, and we plotted the point on here. And anyone who was above the normal range, so they were in that yellow block or that red block, we went away and PCR validated their repeat expansion. And if the colour of that dot represents whether or not the PCR validation came back as intermediate sized or larger pathogenic size. So you'll see if you look at SCAR8, there is one red dot amongst the yellow block. That was an intermediate size expansion that we picked up in someone using the bioinformatics tool. And then when we PCR validated it, we actually found out that it was much larger than that, and that individual had a pathogenic or a repeat expansion that's pathogenic for spinocerebellar ataxia type 8. So there's a lot of individuals with these expansions in our cohort, and we found that just under 2% of our patients had a repeat expansion that's causal for another neurodegenerative disease. So this includes SCAR1, SCAR8, Fragile X syndrome, and a couple of myotonic dystrophies. So we have individuals with pathogenic expansions that usually cause these diseases, yet they've got motor neuron disease. And we went away and we had a look at the clinical notes for all of these individuals, and they had all been seen by a neurologist, and we were wondering whether or not any of these individuals had been misdiagnosed or anything like that, but the clinical notes were consistent with a diagnosis of motor neuron disease. And so it was quite interesting to note this as to whether or not these pathogenic repeat expansions are risk factors for disease in these individuals, or why we're finding them in individuals with motor neuron disease when they usually cause a different kind of neurodegenerative disease. So this isn't too surprising, but the repeat expansions in C9orf72 and ataxin-2 were more common in cases versus controls, and this is known in the literature, so this wasn't a new finding, but this confirmed a lot of previous studies that repeat expansions in these two genes are either causal or associated with motor neuron disease. And we did find some very rare expansions in our cohort. They weren't necessarily associated with disease, so they didn't reach statistical significance. But for example, the Huntington's, we had 2 individuals with an intermediate size Huntington expansion, and that was 2 times more common in our cases versus the controls. And we found individuals with pathogenic mutations that cause myotonic dystrophies, and either one or no controls. And again, we were looking at 600 or so cases versus 5,000 controls. So it's really quite surprising that you're finding some cases with pathogenic expansions here and almost no controls. And one more finding was that intermediate expansions in ataxin-1 that usually cause spinocerebellar or associated with spinocerebellar ataxia type 1, they weren't associated with motor neuron disease in our cohort, when there's some literature that's sort of suggesting that these expansions are associated with motor neuron disease. And we found about 12% of all of our cases and all of our controls had this expansion. And so whether or not this is truly associated with disease risk or not, and at least in our study, we did not find that to be the case. So those sort of, we screened 21 sort of known neurodegenerative disease associated repeat expansions, but since then there has been a few more that have come out in the literature, and people are claiming that they're associated with motor neuron disease. So here's a couple, one in a gene called NEC1 and another in STMN2. We screened our cohort for these exact repeat expansions, and we did not find that these expansions were associated with MND in our Australian cohort. So there's a lot of research, really new research has been put into this area because it's been quite hard to do these kind of analyses to date. And we've had some quite interesting findings, and there's things that might be on the cusps of being interesting, for example, the myotonic dystrophies. But potentially we need to expand our cohort to be able to find if these are actually truly associated with disease, or if we're just finding things that might be prevalent in the population. So I'm just going to leave those results there, and I would really like to thank the MND patients and their family members who have contributed to our studies and the biobanks that have really allowed us to be able to do all of this genetic research. Without that, we wouldn't be able to do any of this research. So it's massive thank you to all of those individuals, and of course the biobanks that collect these samples and allow us to do research with them. And of course all of our national and international collaborators. We've had a lot of people work on these projects with us, and this wouldn't be possible without all of the funding bodies that of course kindly provide funding for us to do this research. So thank you, and if there's any questions, please ask away.
Thank you, Lyndal and Kelly, for your insightful presentations on gene discoveries relevant to M&D. We'll open the digital floor to questions by please click on the raise button on the top panel so we know you have a question. And also remember to, if you can, turn on your camera and introduce yourself. So we'll just wait for a few questions to come up, but I'll have a question firstly a general question to Kelly or Lyndal. On your 25 years of gene mutation screening, your majority of C9 orphan SOD1 up to about 55–56%, and then there was a large unknown. The large unknown is, again, general question on predictions on how many genes you think are involved there in that unknown section, and the time perhaps to identify them.
Oh, well, as I mentioned, I've been in this space for a long time, and this has frustrated me, these remaining, third of patients, because they're missing out on so many of the benefits. The thing is, it's actually some work that we're currently doing that's funded by FightMND. So hopefully we'll be able to talk about it next year after we've published it, looking to put some of these families together using cryptic relatedness to actually be able to find these mutations. We don't suspect that there would be anything as large as what we found for C9orf72 or for SOD1. There may be more likely to be private mutations, so mutations that occur only in a single family. But even if we find lots of these mutations, it's the biological mechanisms that we understand from finding these mutations that will help us build a better picture of MND. So it's, whether the timeline, look, when exome sequencing first was introduced in 2009, I remember at one of the international ALS conferences, we sat down with all the big genetics people around the world, and we all thought we'd be out of a job within 5 years because everything would be solved, and it hasn't happened. It hasn't. Even though we've had all these advances in technologies, it's really the types of samples that we need to find new mutations. You need families with more than one affected individual, because with singletons, it's more a statistical exercise rather than actually being able to find definitive causal genes.
Thank you. We have a question from Cam Wells. Cam, we'll ask you to come on. Thank you. And unmute if you can.
Like you should be.
Technicalities.
Would you like to just put your question into the chat? Sorry.
Question to Lyndal while we're waiting on short-term and repeat expansions, obviously. You're looking at predominantly the known tandem repeats. Have you gone into looking at, or any insight into looking at, novel tandem repeats, or is that something that you're looking to pursue?
Yeah, that's a really good question. So we have started looking at de novo short tandem repeats. There's not as much, so this is a very new field of research, and so while there are bioinformatics tools that can pick these up, what you do with them when you find them and how you find the overrepresentation in cases versus controls is a little bit more difficult. So the techniques for refining these once you've found them, it's not so straightforward as it sounds like it should be, but it's not. So we are working on it, but we don't have anything to show at this stage for that work, but it is being done.
And I might add to that as well. So Lyndal's mentioned de novo repeats. There's currently, in the human genome, there's around 174,000 actual short tandem repeats. So theoretically, any of those could be expanded. And, you know, if we were looking at, how many did we look at? 20? 20 across, say, 5,000 cases and controls. This would then be looking at 174,000 across 5,000 people. So now the compute resources that are required are massive, as well as the statistical analysis to pull out what's actually real.
We've got the question from, thanks Kelly and Lyndal. We've got the question from Cam Wells. Do you think the missing inheritance is from single genes or more interrelated network effects, that is oligogenic, polygenic effects?
I would say in terms of sporadic form of disease, yes, I think we're looking at more, I like how you've described it, interrelated network effects. I think, yes, we for sporadic disease, we would be looking at more combinations of genes, which is in other research fields, a bit in motor neuron disease, but not as developed in other research fields, is looking at polygenic risk scores. So actually looking at the gene variants that you have and assigning a risk score to an individual based on the genetics they have. In terms of the familial form of the disease, where there's missing third, I think it may be both. It may be both. We may be having definitive causal mutations, but there may be some cases where oligogenic inheritance might be either reducing penetrance or increasing penetrance.
Thank you. I'll throw it back to Sarah.
Great. Thanks, everyone. Thank you, Kelly and Lyndal, for such a wonderful and informative presentation and a fantastic discussion. It was great to hear about your recent publication, Lyndal, and about how both your work is contributing to an exciting era in M&D genetics. So if there's anything else that anyone would like to follow up with our speakers today, you can get in touch directly with them or get in touch with us at FightMND if you would like us to help connect you. Today's seminar has been recorded and we will be available on our website in about a week's time. So if you know of any of your colleagues who weren't able to join live today, please let them know that they can watch it soon. So thank you again for tuning into the FightMND Scientific Seminar today. We hope that this series has been creating new opportunities and ideas for our research community. The August seminar will be held on Tuesday, August 29th at 12 p.m. So keep an eye out on your inboxes for details of next month's speaker. Until next time, thank you and see you again soon.